Hello, so today we'll be discussing Chapter 2 of Anatomy and Physiology. This is on the chemical basis of life. So, the smallest, most fundamental material component of the human body are our basic chemical elements. In fact, chemicals called nucleotide bases are the foundation of the genetic code. And we know the genetic code is the instructions of how to build and maintain the human body from conception all the way up to old age. So, why do we study chemistry in A&P? Human chemistry includes our organic molecules, which are our carbon-based molecules, as well as our biochemicals. And those are all the chemicals that are produced by the body. Human chemistry also includes elements, and we're going to talk about elements throughout the entire PowerPoint. In fact, life cannot exist without many of the elements that are a part of our Earth. Elements that include phosphorus, carbon, sodium, and calcium, just to name a few. These elements, in turn, will form both our inorganic and our organic chemical components that are important to our life. These type of chemical compounds are our water, our glucose, and our proteins. Now let's talk about the fundamentals of chemistry. Scientists define matter as anything that occupies space and has mass. Everything, all of the substances of the universe, from a grain of sand all the way up to a star in the sky, is what we call matter. An object's mass and its weight are related concepts, but not quite the same. So an object's mass is the amount of matter that is present. Okay, it's the amount of matter that is present and is the same whether the object is on Earth or is in zero gravity environment in outer space. Okay, on the other hand, an object's weight is its mass or heaviness as it is affected by the pull of gravity. So gravity is involved when you're talking about weight. An object's weight is greater where the pull of gravity is stronger than where the gravity is less strong. So, for example, an object of a certain mass weighs less on the moon than it does on Earth because the gravity on the moon is less than it is on Earth. In other words, weight is variable. Okay, it changes and it is influenced by gravity. So, a piece of cheese that weighs a pound on Earth weighs only a few ounces on the moon, okay? So those are three major definitions that you're going to need to know individually as well as how they relate to one another. Now let's talk about the definition of chemistry. So chemistry is just the study of composition, properties, interactions of matter, while biochemistry is our biological chemistry. So it studies our physiological processes within the body as well as disease. All matter in the natural world is composed of one or more of the 92 fundamental substances, and these substances are called elements. An element is a pure substance that is distinguished from all other matter on Earth by the fact that it cannot be created or broken down by any of our ordinary chemical means, okay? You can't burn it, you can't freeze it. Elements are going to be the pure substance. There are 92 naturally occurring elements that we have identified. In nature, elements are rarely going to occur alone. So if they don't just live on their own, instead, they are going to be combined with one another in order to form what we call compounds. Okay, so our elements are going to combine to form compounds. A compound is a substance that is composed of two or more elements, and it's usually joined by a chemical bond. For example, the compound that we call glucose that we have in our body, it's a very important fuel for our body. It is always going to be composed of the same three elements: carbon, hydrogen, and oxygen. Okay, so that's a compound. Now an atom is the smallest quantity of an element that retains its unique properties of that element. Okay, it's the smallest type. In other words, an atom of hydrogen is a unit of hydrogen. It is going to be the smallest amount of hydrogen that can exist in the earth. Okay, So when we refer to an atom of hydrogen, it's going to be the smallest particle of hydrogen that can ever exist. So as you might guess, atoms are almost unfathomably tiny. They're teeny, teeny, tiny. The period at the end of a sentence is 1,000,000 times bigger than an atom, okay? So that puts it in perspective for you. So different elements are required by the body in different amounts. So let's talk about what we name these elements. So when we're referring to bulk elements, these are elements that are required by the body in large amounts. So bulk elements are going to be your carbon, your oxygen, your hydrogen, your nitrogen, your sulfur, and your phosphorus. Okay, so these are your bulk elements. Now your trace elements are elements that are required by the body in small amounts. They are small amounts. So elements such as your iron and your iodine. And then we have something called our ultra trace elements. These are required by the body in very minute amounts, so even smaller than the trace elements. They are teeny tiny amounts needed. So arsenic. Okay, so those are going to be different types of elements that are required in different amounts. So it's important that you know how we classify each of those. All right, so atoms, we talked about atoms, how they are tiny, tiny particles, but these atoms are made up of even smaller subparticles, okay, which include three important types. We have protons, neutrons, and electrons. Now our table 2.1, as you see here, is going to break it down for us. So let's take a look. The first thing is an atom. What's a characteristic of an atom? We know it is the smallest particle of an element that has properties of that same element. Remember the hydrogen atom. Okay, it is a unit of hydrogen, therefore it is the smallest amount of hydrogen that can ever exist. Okay, that is a hydrogen atom. Following on to the next part, we're going to name it a neutron. So these neutrons are relatively large particles that are within the same atom, okay? They are uncharged, and thus we declare them as electrically neutral, okay? These are found within the atomic nucleus, okay? So the next one is our electrons. Our electrons are these extremely small particles, they're tiny, that are going to be found within the atom as well. They almost have no weight, and this is very important. They carry a negative electrical charge. Electrons have a negative electrical charge, and they are in constant motion around the atomic nucleus. Okay, so they are orbiting around the atomic nucleus. Now, our protons are relatively large particles, just like our neutrons, but they carry a positive electrical charge. Protons carry a positive electrical charge, and they, just like the neutrons, are found within the atomic nucleus. Okay? All right, so now we're going to talk about some more of the major elements and traces within the human body. But first, I want to refer to, we don't have a picture here, but we have something within the scientific world called the periodical tables or periodic table of elements. This is a chart that is identifying the 92 elements that are found. The periodic table of elements is a chart that identifies the 92 elements that we talked about at the beginning of the PowerPoint that are found within nature, as well as several larger and unstable elements that are discovered by scientists experimentally. These elements are arranged in order of their atomic number, with hydrogen and helium elements at the top of the table and the more massive elements toward the bottom. So, now that we talked about the periodic table, let's talk about table 2.2 in your book. This is going to show some of the more major and the trace elements within the human body, what we have in our body. And these are going to be sorted by weight. So elements within the body that require large amounts, that the body requires large amounts, are going to be our carbon, our hydrogen, our oxygen, our nitrogen, our sulfur, and our phosphorus. And remember, these are termed as our bulk elements, And then the elements These elements are going to be making up more than 95% of the human body. Also, on this table, you'll see elements that are required in small amounts within the human body, and these are what we refer to as trace elements. And these elements are going to be your cobalt, your copper, your fluorine, your iodine, your iron, and your zinc. So let's refresh our memory a little bit. We said that atoms are made up of even smaller subatomic particles, and these particles are known as protons, neutrons, and electrons. In atoms, protons and electrons are going to carry electrical charges. Remember, our large proton particles carry a positive charge and are designated by a P. The extremely small particles that we know are known as the electron. These have a negative charge, and they are designated by an E. And in atoms, neutrons, which are considered relatively large in size, just like our proton, these have no charge, okay? These have no charge. They are electrically neutral. So the number of positively charged protons and non-charged or neutral neutrons are going to give us the mass to the atom. And they're going to make up the central nucleus, okay? The central nucleus. So your protons and your neutrons are going to be the central nucleus. They're going to make up that. While our negatively charged electrons are spinning around the nucleus, close to the speed of light, okay? Close to the speed of light. So just like how a magnet sticks to a steel refrigerator, because you have their opposite charges are going to attract, your positively charged protons are attracted to your negatively charged electrons. This mutual attraction is going to give the atom some structural stability. The attraction by the positively charged nucleus is going to keep our electrons from straying off. The number of protons and electrons within a neutral atom are usually going to be equal. That is very important. They're going to be equal. Thus, that is going to give your atom an overall charge that is called balanced, okay, or electrically neutral, okay? So when your protons equal the number of electrons, you then have an electrically neutral atom. Figure 2.1 is a picture of an atomic structure, okay? This is an atomic structure of an element that we call lithium. It shows a central nucleus with what two subparticles? You have neutrons and protons. So this one in particular is showing 4 neutrons, indicated by the neutron, the 0, which is going to be neutral, as well as 3 protons. Those are going to be your orange subparticles. There are 2 orbitals around the nucleus where you will find our electrons. We have 2 electrons in the inner orbital. 1, 2. And 1 electron in the outer orbital. And this is the 1. Now let's talk about atomic number and mass number. The atomic number, which is the number of protons within the nucleus of an atom, identifies our elements. Since an atom usually has the same number of electrons as protons, remember those are usually equal, the atomic number is going to identify the usual number of electrons as well. So when you look on the periodic table of elements and you see the atomic number, that will tell you how many protons is in that element and how many electrons is in that element. The element's mass number, on the other hand, is the sum of the number of protons and neutrons within the nucleus. Okay, it is the sum, the adding of your protons and your neutrons together within the nucleus. That is your element's mass. Although each element has a unique number of protons, it can exist as different isotopes, okay? An isotope is one of the different forms of an element. It is distinguished from one another by the different number of neutrons. So how you can tell an isotope from the regular element is that you'll have a different number of neutrons. An isotope that contains more than the usual number of neutrons is referred to as a heavy isotope. Okay, it is a heavy isotope. An example is 14C. Okay, 14C, that is a carbon with more than the regular number of carbons. A heavy isotope tends to be unstable, and an unstable isotope is referred to as radioactive. A radioactive isotope is an isotope where the nucleus readily decays. Okay, it's giving off subatomic particles and electromagnetic energy. So excessive exposure to radioactive isotopes can damage our human cells and even cause cancer or birth defects. But when you are exposed to these radioactive isotopes in a controlled environment, some radioactive isotopes can be used for medicinal purposes. They are used in medicine. So let's talk about ionizing radiation. Radiation, whether it's alpha, beta, or gamma, is called ionizing radiation because its energy can remove electrons from the atom, resulting in the formation of ions. The free electrons can damage nearby atoms. Ionizing radiation sources include X-rays, naturally occurring radioactive elements in the crust of the earth, and our nuclear weapons. Okay, so those are some sources where you'll find ionizing radiation. Now let's talk about molecules, compounds, and molecular formulas. What is a molecule? A molecule is defined as a particle that is formed when two or more atoms chemically combined. Okay, so atoms make up molecules. What is a compound? A compound is when a particle is formed when two or more atoms of different elements chemically combined. So make sure you understand the difference between a molecule and a compound. A molecule is when two of the same element combine to form a particle, while a compound is going to be, remember, two or more of an atom, of different elements, okay? Now, what is a molecular formula? A molecular formula is there to depict the elements present and the number of each atom that is present in that molecule. So look at some of the molecular formulas listed here. H2, hydrogen, is a molecule of hydrogen. C6H12O2 is a molecular formula. It is a molecule of glucose found within the body. H2O is a very popular one. That is a molecular formula, for the molecule of water. So, if atoms of the same element combine, they produce molecules of that element, okay? Of that element. Now, if atoms of different elements combined, then you're going to have molecules that form a compound. Okay, that form a compound. So let's take a look here at this figure. Two atoms of hydrogen, for example, can combine with one atom of oxygen to produce a molecule of a compound of water. Okay, just as this figure shows, 2.2 shows. Also, table sugar, baking soda, natural gas, and most drugs are known as compounds, okay? Why? Because you have two or more of different elements combining, okay? Two or more of different elements combining. So take a look. This is what we refer to as a molecule. This is a molecule. While these are compounds, okay? You see different elements have combined. The same element has combined here. So that's how we would know the difference between a molecule and a compound. So let's talk about some bonding of atoms. Chemical bonds form when atoms combine with other atoms. They result in interactions between the electrons of these atoms. Electrons of an atom occupy regions of space called electron shells, okay? And these electron shells are what we saw that encircle the nucleus, okay? So what you see those electrons floating on, those are what we refer to as electron shells. For atoms with atomic numbers of 18 or less, the following rules apply. The first shell can hold up to 2 electrons. The second shell can hold up to 8 electrons, and the third shell can hold up to 8 electrons as well. The lower energy shells, or the inner orbits, are going to always be filled first. So the ones closest to the nucleus are going to be filled first and are the most stable with a certain number of electrons in the outermost shell. Because remember, it's 2, 8, okay? Or you can have 18 in larger atoms, okay? So we're looking for stabilities. Electrons in the outermost shell are going to determine whether an atom will react with other atoms in order to form chemical bonds. Chemical bonds. So you're looking at the outermost shell when you want to bond with another atom. Okay, so figure 2.3. These are electrons that orbit the atomic nucleus. So the structure of hydrogen shows a central nucleus with just one proton. Okay, that's the orange, one proton. Hydrogen has an orbital with one electron. You see the little blue small particle on the first shell. It just has one electron. Now, the structure of helium shows a central nucleus with 2 neutrons and 2 protons. Helium has an orbital with 2 electrons. So you're going to see 2 electrons floating in that most inner orbital. 1, 2. The structure of lithium, okay, we looked at this one earlier. The structure of lithium shows a central nucleus with 4 neutrons and 3 protons. There are 2 orbitals with 2 electrons. Remember, 2 is the max for the first orbital. There are 2 electrons in the inner orbital and 1 electron in the outer orbital. The structure of lithium shows a central nucleus with 12 neutrons and 11 protons. There are 3 orbitals here. 3 orbitals with 2 electrons in the inner orbital, 8 electrons in the middle orbital, and 1 electron in the outer orbital. The atomic number is 11, and the atomic weight is 23. Before we move on to the next slide, I want to correct myself. I said lithium for the second figure here. That is a structure of sodium, not lithium. Sodium has 12 neutrons and 11 protons, okay? Alrighty, so an atom that has an electrical charge, whether it's positive or negative, is an ion. A potassium ion is written with a K, indicating that it has lost a single electron. A positively charged ion is known as a cation. The ionized form of fluorine is called fluoride and is written as an F minus. A negatively charged ion is known as an anion. The opposite charges of cations and anions exert a moderately strong mutual attraction that keeps the atoms in close proximity, forming what we call an ionic bond. An ionic bond is an ongoing close association between ions of opposite charges. The table salt you sprinkle on your food owes its existence to ionic bonding. So table salt, NaCl, is a very well-known and popular ionic bond. So let's take a look at figure 2.4. Step 1 is the formation of ions. So sodium has 11 protons, 11 electrons, and 12 neutrons, while our chlorine atom has 17 protons, 17 electrons, and 18 neutrons. An electron on the outermost orbital of the sodium points to the outermost orbital of the chlorine atom, which brings us to step 2. Step 2 is the attraction of those opposite charges, okay? So in which the sodium ion is positively charged after donating an electron to the chlorine, is negatively charged after accepting the electron from the sodium ion. So now the number of electrons in the sodium are 10, and in chloride ion, you're going to have 18, okay? The sodium donated one to the chloride. Step 3 shows the formation of our ionic compound, okay, in which the sodium and the chloride ions are going to combine to form what we know as sodium chloride. Sodium chloride. And then B here in this figure is just the attraction of the many sodium and chloride ions that's going to result in the formation of a large grouping which we refer to as a crystal. Okay, as a crystal. This is a space-filling model of sodium chloride crystal. Sodium chloride crystal. Next up are going to be our covalent bonds. So unlike our ionic bonds that are formed by the attraction between a cation's positive charge and an anion's negative charge, molecules are formed by a covalent bond, which they share electrons in a mutually stabilizing relationship. Share is the key word here when you're talking about covalent bonds. So like your next door neighbor whose kids want to hang out first at one home and then at the other home, the electrons do not lose or gain electrons permanently. Okay, not permanently, they are shared. Instead, the electrons are going to move back and forth between the elements because of the close sharing of the pairs of electrons, one electron from each of the two atoms. The covalent bonds are usually much stronger than your ionic bonds. Okay, they're much stronger than your ionic bonds. Figure 2.5a is an illustration that shows two hydrogen atoms, each with one proton and one electron. They form a covalent bond to form a hydrogen molecule by sharing the one electron. Okay, they are sharing the one electron. That is a covalent bond. This illustration is going to show two hydrogen molecules and an oxygen molecule that form a covalent bond to form two water molecules, okay? So an oxygen is going to share an electron with each of the two hydrogen atoms to form the water molecules, okay? To form the water molecules. Structural formulas. Structural formulas are going to show how atoms bond and how they rearrange or arrange in various molecules. One line between the atom means that one pair of electrons are being shared. Okay, so if you see one line, that is one electron being shared, forming a single bond, while two lines indicate that there are two pairs of electrons being shared. And that's what we call a double bond. They form a double bond. And we will see on the next slide exactly what that means. So take a look here. You have two hydrogen atoms. They have one line here, okay? So they are sharing one pair of electrons and they are forming a single bond. While these two oxygen atoms are forming a bond, but they are forming a double bond, and this is indicated by two lines. Two lines. So remember, in covalent bonds, electrons in the outermost valence shell are shared with the valence shells of both atoms, ultimately stabilizing both of the atoms involved. So in a single covalent bond, a single electron is being shared between the two atoms. And there are even triple covalent bonds where there are three atoms shared, okay? So let's talk about nonpolar and polar covalent bonds. In nonpolar covalent bonds, the sharing of the negative electrons is relatively equal, as is the electrical pull of the positive protons in the nucleus of the atoms involved. This is why covalent bonding molecules or bonded molecules that are electrically balanced in this way are described as nonpolar. That is, there is no region of the molecule that is either more positive or more negative than the other. It's a good balance, okay? Now, polar covalent bonds occur when atoms share electrons unequally, okay? They are not shared equally. They are found between the atoms with different electron negativities. They form polar molecules with unequal charge distribution. Atoms with larger number of protons have a higher electron negativity, and then they pull the shared electrons closer to their nucleus. So it's not equal, it's unequal. Water is a polar molecule. We're going to talk about that here in a minute. So in chemistry, a polar molecule is a molecule that contains regions that have opposite electrical charges. Polar molecules occur when atoms share electrons unequally. Okay, remember, it's unequal in especially in our polar covalent bonds. The most familiar example, which we stated before, of a polar molecule is water. Water, that is, the oxygen region has a slightly negative charge, okay? Slightly negative charge. In the region of the two hydrogen atoms have a slightly positive charge. The opposing electrical charges of the molecule are going to pull that mass in such a way that it creates a shape somewhat like a triangular tent, okay? Look at the triangular tent here. With the positive charges at one end that are formed by the hydrogen atoms, those are going to be at the bottom of the tent. And the negatively charged at the opposite end make up the charged regions highly likely to interact with the charged regions of the polar molecules. So for human physiology, it's important to note that the resulting bond formed by the water is one of the most important, okay? And this bond is what we refer to as a hydrogen bond, okay, which we will discuss next. Hydrogen bonds. A hydrogen bond is formed when a weakly positive hydrogen atom already bonded to one electronegative atom, for example, the oxygen in the water molecule, it is going to be attracted to another electronegative atom from another molecule. In other words, hydrogen bonds always include hydrogen. That is already part of a polar molecule. So the most common example of hydrogen bonding is the natural world. In the natural world, occurs between molecules of water. Molecules of water. So it is known as a relatively weak attraction, but it is very important within our body. Hydrogen bonding occurs because the weakly negative oxygen atoms in one water molecule is attracted to a weakly positive hydrogen atom in two other water molecules. So hydrogen bonds, like we said, are relatively weak, and therefore they are indicated with a dotted line. As you can see in figure 2.8b, you see this dotted line. We are using that to indicate that it is a relatively weak bond rather than a solid line like you'll see in some other illustrations. A solid line would indicate a stronger bond. Chemical reactions. So chemical reactions occur when chemical bonds form or break between atoms, ions, or molecules. So these reactions are going to form larger molecules from our smaller molecules or atoms, or when bonds between the components of larger molecules break, releasing our smaller molecules or atoms. So a chemical reaction can go both ways. Okay, it goes both ways. All chemical reactions begin with what we call a reactant. A reactant is the general term for one or more substances that enter into the reaction. Okay, that is a reactant. Now, one or more substances that are produced by a chemical reaction, the end product, is what we refer to as the product. As the product of a chemical reaction. Now let's talk about the different types of chemical reactions. A synthesis reaction is a chemical reaction that results in the synthesis or joining of the components that were formerly separated. That is, more complex chemical structure is formed, okay? More complex chemical structure is formed. The general equation, as you can see here, is going to be A plus B yields AB. Two component bonds to make one large molecule. So A and B are going to be the two components that are going to bond together to form a larger component. Or molecule, excuse me. The next one is decomposition reaction. This is going to be also a chemical reaction, but this one is going to break down or decompose something that is larger into its constituent parts, okay? So the general equation for a decomposition reaction is going to be AB, so a larger part that is broken down into two individual constituents. So you have AB yields A plus B. Now, the next one is our exchange reaction. this is going to be a chemical reaction in which both synthesis and decomposition occurs, okay? So you have chemical bonds that are both formed and broken within this reaction. As a result, your components of the reactants are going to be rearranged, okay? Because you have some synthesis occurring as well as some decomposition occurring. So the equation is going to be AB plus CD is going to yield ADCB. So you see, our reactants were just rearranged. Reversibility is also a quality of exchange reactions. A reversible reaction is when the products can change back into your reactants. A plus B can yield AB, and then AB can yield A plus B. So they go back and forth. That is reversibility reaction. Okay, so electrolytes, acids, and bases, and salts. First up are electrolytes. Electrolytes are substances that release ions within water. A solution can conduct electric current, so it's called an electrolyte. Here is our example. NaCl yields Na plus Cl. Acid and bases, like our salts, are dissolved in water into electrolytes, okay? They dissolve into water into electrolytes. So our electrolytes make up our acids and our bases. Acids and bases can very much change the properties of the solution in which they are dissolved, okay? An acid, first up, is a substance that releases hydrogen ions into a solution. Our strong acids are compounds that have released all of their hydrogen atoms into the solution. So that is considered a strong acid. That is, they ionize completely. They are completely ionized. They have released all of their hydrogen atoms. Hydrochloric acid, HCl, which is released from the cells within the lining of our stomachs, is considered a strong acid because it releases all of its hydrogen atoms into the stomach's watery environment. This strong acid aids in our digestion and kills ingested microbes. Very important. Now our weak acids do not ionize completely, okay? That is, some of their hydrogen ions remain bonded within the compound in the solution. So an example of a weak acid is vinegar, okay, or acetic acid. It is called acetate after it gives off one proton. Now, a base. A base is a substance that releases hydroxyl ions, OH, into a solution, and one that will accept hydrogen ions already present within a solution. So our strong bases are going to release most or all of their hydroxyl ions, while our weak bases are going to release only some of our hydroxyl ions and absorb only a few of our hydrogen ions. So moving on to our salts. You can define a salt as a substance that when it's dissolved in water, it dissociates into ions other than hydrogen or hydroxyls ions. Okay, so other than our acids and our bases. These ions are considered electrolytes formed by a reaction between an acid and a base. They are capable of conducting an electrical current in a solution. So this property is critical to the function of ions in transmitting nerve pulses within our body and prompting our muscle to contract. Okay, so they are very important, these salts. So a little more on our acids and bases. The study of acids and bases are involved in measuring, like we said, our hydrogen ions concentration within a solution. So when an ionically bonded substance is put into water, these charged ions are attracted to a slightly charged end of a polar water molecule. And it's going to begin to dissociate. This dissociation of the substance and the ions become surrounded by water molecules. And this substance is now what we refer to as an electrolyte, since it can now carry an electrical current. So here's our crystal. Remember, we see multiple of elements formed here. We refer to as a crystal. So this is a crystal of sodium chloride, salt crystal. This is shown in a space-filled filling model with all of our sodium and our blue sodium ions, and our green chloride ions. So this salt crystal is placed into water, and you'll see the dissociation of sodium and chloride ions within the water. It is represented by the water molecules that are around the sodium and the chloride ions. So this is how it associates within water. The relative acidity or the alkalinity, which we know as the basic side of a solution, can be indicated by its pH. Okay, so now we're going to talk about pH a little bit. A solution's pH is the negative base 10 logarithm of the hydrogen ion, which we refer to as H+, concentration within a solution. Okay, the concept of pH will begin to make more sense when you study the pH scale. Okay, the pH scale consists of a series of increments ranging from 0 all the way up to 14. A solution with a pH of 7, right in the middle, is considered neutral. Okay, it is considered neutral. It is neither acidic nor basic. Pure water has the pH of 7. Okay, it is a neutral. It has a neutral pH. The lower the number, so below 7, below water, the more acidic the solution, okay? The more acidic solution. Also, the greater the concentration of our hydrogen ions. The higher the number, so above 7 to 14, means the more basic a solution is, or alkaline the solution is. Or you also can refer to as it has a lower concentration of our hydrogen ions. Human urine, for example, is 10 times more acidic than our pure water, okay? And HCl, our hydrochloric acid, is 10 million times more acidic than water, okay? So those are going to be found toward the bottom, 1 to 7. They're going to be more acidic. So table 2.5 shows you the relationship between the hydrogen ions concentration and the pH that we just spoke about. So the concentration of hydrogen ions at each pH value is 10 times different than the next pH, okay? So 10 times different, 10 times different, 10 times different as you go up, okay? For instance, a pH value of 4 corresponds to a proton concentration of 4 to the negative 4th power. Okay, while the pH of a value of 5 corresponds to the proton concentration of 10 to the negative 5 power. And you can see the higher the number above 7, the more basic. Okay, above 7 is more basic or alkaline the solution. In the lower concentration of hydrogen atoms. Okay, so we have a lower amount of hydrogen atoms. This is a small number. While the lower number, so anything below 7 and down, is going to be a more acidic solution. These also have a greater concentration of hydrogen atoms or ions. Here are the definitions of the terms that we have discussed thus far. So pH scale, it indicates the concentration of our hydrogen ions in a solution. Neutral is going to be when we have a pH of 7. Okay, of 7. It indicates equal concentrations of hydrogen ions as well as our hydroxyl ions. This is our pH of pure water, like we said before. Acidic is when you have a pH that is less than 7. It indicates that you have a greater concentration of our hydrogen atoms than our hydroxyl atoms. Now, our basic or alkaline pH is when it is greater than 7, so 7 and up. 7 and up indicates that it is an alkaline or basic solution, and indicates that it has a higher concentration of hydroxyl ions than our hydrogen ions. Figure 2.10, this is an actual pH scale. It illustrates the pH values of some of our common substances, okay? So as a reminder, 7, once again, a pH of 7 is neutral. is neutral. You see, it's indicated by distilled water. pH 0 to 7 is acidic. 0 to 7 is acidic. And 7 to 14 is basic or alkaline. We see at the bottom of the illustration, those arrows are going to indicate that the hydrogen ion concentration is going to decrease from the pH 1 to 7, excuse me. So you see hydrogen ion concentration is increasing or decreasing, excuse me, from 7 to 1, because you're going to get more acidic as you go down. So let's look at some of the pH values here. Our pH values of gastric juice is 2. Apple juice is 3. Our tomato juice is 4.2. Cabbage is 5.3. Corn is 6. So it's considered acidic. Cow's milk is slightly acidic, is 6.6. Distilled water, once again, is our neutral, 7. Human blood is slightly basic, okay? Slightly basic. It is 7.4. Egg whites are 8. Sodium bicarbonate is 8.4. Milk of magnesia is 10.5. And our most basic on this scale is going to be 11.5. That is household ammonia. What people use to clean. The pH of human blood normally ranges from 7.35 to 7.45, although it is typically identified as 7.4, like we saw on the chart here. At this slightly basic pH, so our blood is slightly basic, blood can reduce the acidity resulting from carbon dioxide, okay? Carbon dioxide within the body, because it is constantly being released into the bloodstream by trillions of cells within the body. Homeostatic mechanisms, finding that balance within the body, along with exhaling carbon dioxide while breathing, this is what helps normally keep the pH of the blood within narrow range, okay? It keeps it at its slightly basic level. This is critical because fluctuations up and down, whether your blood is too acidic or too alkaline, can lead to a life-threatening disorder. Okay, so these disorders we refer to as acidemia or alkalemia. Acidemia is going to be when the blood pH drops to 7, okay? It goes down. It goes 7 through 7.3, okay? So it becomes more acidic. Acidosis makes a person feel disoriented and fatigued. It's caused by vomiting of alkaline intestinal contents. Diabetes can cause it, lung disease, when you have impaired carbon dioxide exhalation. Okay, so that's going to be one of the disorders. Alkalemia is going to be when it is going to occur when your pH rises above the normal level. So it's in a range of 7.5 to 7.8. Alkalosis also makes a person feel dizzy and agitated. It is caused by high altitude breathing, vomiting of acidic stomach contents, high fever, or taking excessive antacids. So all of the cells of the body are going to depend on this homeostatic regulation of your acid-base balance. You see, you need balance within the pH, okay? So the acid-base balance of the pH at approximately 7.4. The body, therefore, has several mechanisms for this regulation. It involves breathing, excretion of chemicals in your urine, the intestinal release of chemicals collectively, which we refer to as buffers into the body fluids. A buffer is going to be a solution of weak acid and its conjugated base. A buffer can neutralize our small amounts of acids or bases in the body fluid. So that's what our buffers are going to do to help find that regulation, that homeostatic regulation. For example, if there is even a slight decrease in your pH, your blood pH, below 7.35 of your bodily fluid, the buffer in the fluid, in this case, is going to act as a weak base. It will bind to the excess hydrogen ions. And then in contrast, if your pH was to rise above 7.45, there's going to be a buffer that's going to come in. It's going to act as a weak acid. That weak acid is going to contribute hydrogen ions in order to bring your pH back down to the normal level. Now let's talk about compounds that are important for the body's structure and function. So in general, these compounds are usually referred to as inorganic or organic. And an organic compound is a substance that contains both carbon and hydrogen. Organic compounds are synthesized via covalent bonds, the sharing bonds within living organisms, including our human body. There are four types of the most important to the human structure and functions, and these are our carbohydrates, proteins, lipids, and our nucleic acids. These are all types of organic molecules. Now let's talk about our inorganic molecules. An inorganic compound is a substance that does not contain both carbon and hydrogen. Usually it dissolves in water and it dissociates, forming ions and are electrolytes. There are four groups of inorganic compounds that we will discuss throughout this PowerPoint. These are considered essential to life. These are our water, our salts, our acid, and our bases. So let's talk about the first inorganic compound, water. So as much as 2/3 of the human body, or the adult body weight, is water. This water is contained both within the cells and between the cells, and this is what helps make up our tissues and our organs. So it contains several roles that make water indispensable to human functioning. Okay, we need water. It's very important. Water is a major component of many of the body's lubricating fluids. So just like we use oil to lubricate a hinge on a door, water is found within our synovial fluid, and this helps to lubricate the actions of our body joints. Watery fluid helps to keep food flowing through our digestive tracts. Water also protects our cells and our organs from physical trauma. It helps to cushion our brain within the skull. For example, in protecting the delicate nerve tissue of our eyes. Water cushions developing fetus within the mother's womb as well. In the body, water absorbs the heat that is generated by chemical reactions without greatly increasing the temperature. So to explain more, when the environmental temperature on the outside is soaring and it's so hot outside, the water stored within the body helps to keep our bodies cool. Okay, so how does it do that? While our sweat glands release warm water in the form of sweat, this water begins to evaporate into the air, and that's going to carry heat away from the body. Okay, carry heat away from the body. In turn, that is going to decrease our body temperature. Okay, it's going to help us cool down. So water has many other important functions, okay? Other important functions. Now, oxygen is used by organelles to release energy from nutrients in order to drive the cell's metabolic activities, okay? It is necessary for survival. Oxygen is necessary for survival. Now let's talk about some inorganic substances. Carbon dioxide. Carbon dioxide is a waste product that is released during metabolic reactions, okay? It must be removed from the body through exhaling, okay? Inorganic salts. These are abundant in body fluids, okay? Fluids. There are sources that are necessary, and these are the ions here on this slide. They play important roles in metabolism. It helps to control our water concentration, our pH, our blood clotting, our nerve, and our muscle processes. You're going to see salts being involved in these. Electrolyte balance exists when gains equal our losses. Okay, so those were our inorganic substances. Now let's switch gears to our organic substances. First up is our carbohydrates. The term carbohydrate means hydrated carbon. Recall that the root word hydro indicates water. So a carbohydrate is a molecule that is composed of carbon, hydrogen, and oxygen. In most carbohydrates, our hydrogen and oxygen are found in the same 2-to-1 relative proportions they have in water. Carbohydrates are referred to as saccharides, okay? A word that means sugar. There are three forms of saccharides that are important to our body. Those are monosaccharides, disaccharides, and polysaccharides. Monosaccharides are monomers of carbohydrates, okay? They have a single sugar. Disaccharides are, di means two, so there are double sugars, and they are made up of two monomers. While our polysaccharides are our polymers, and they consist of hundreds to thousands of monomers, which is why we refer to it as our complex carbohydrates. So, examples of our monosaccharides are going to be glucose and fructose. Disaccharides are going to be sucrose and lactose. And then our complex carbohydrates, what we refer to as our polysaccharides, are our starches, our glycogen, and our cellulose. So let's look at figure 2.11, our carbohydrates. So some glucose molecules, remember our formula C6H12O6, have a straight chain of carbon atoms. One terminal C atom is bonded to an O oxygen atom and a hydrogen atom. Another carbon atom is bonded to two hydrogen atoms and a hydroxyl group. That's the OH. That's the OH. The other carbon atoms are going to be bonded to a hydroxyl group and a hydrogen atom each. Let's look at B. More commonly, glucose molecules form a ring structure, okay? They're going to form this ring structure. The substituents of each carbon atom are shown here, okay? So that's going to be your ring structure. And then part C, this is going to be a shape that symbolizes the ring structure of a glucose molecule, but it is the same. It just doesn't have all of the molecules written in. It is a 6-carbon ring with one oxygen atom. So figure 2.12, this illustration A there where it says monosaccharide shows a monosaccharide that has 5 carbon rings that are fused together to an oxygen atom, okay, just like we saw previously. Now B is a disaccharide. It shows a disaccharide that has 2 monosaccharides bonded to each other, okay, by an oxygen atom. And then C is our complex carbohydrate, our polysaccharide, and this has chains of multiple monosaccharides. Now we're going to move on to lipids. A lipid is one of the highly diverse groups of compounds made up mostly of hydrocarbon. Their nonpolar hydrocarbons make all lipids hydrophobic. Okay, they are afraid of water, water-fearing. In water, lipids do not form a true solution, but they may form an emulsion, which is a term for a mixture of solutions that do not mix well, okay? So it does not mix. Therefore, they are considered insoluble in water, okay? They do not dissolve in water. Lipids do not dissolve in water. A triglyceride is one of the most common dietary lipid groups and the type found most abundantly within our body tissues. This compound, which is commonly referred to as a fat, okay? Now, let's talk about phospholipid. A phospholipid is similar in structure to a triglyceride, but our phospholipids are said to have hydrophobic tails, okay, so water-fearing tails containing neutral fatty acids, and they have hydrophilic heads. Hydrophilic means water-loving, so they have heads that enjoy or love water. And they have a nitrogen atom and a phosphate group attached. Okay, so that's going to be the structure of a phospholipid. Now let's talk about a steroid compound. A steroid compound is the most important contribution to the human structure, and the function is what we know as a cholesterol. Okay, a cholesterol. This is synthesized by the liver in humans and animals, and is also present in most of our animal-based foods. Cholesterol is also what we refer to as a building block of many hormones, signaling molecules that the body releases to regulate processes at a distant site. So finally, like our phospholipids, cholesterol molecules are found within the cell membrane. Okay, they are important. They are found within the cell membrane, where their hydrophobic and hydrophilic regions help to regulate the flow of substances into and out of the cell. Okay, so they're going to help with functioning of the cell membrane. So now we're going to talk about our saturated fatty acids and our unsaturated fatty acids. So our saturated fatty acids have only single carbon to carbon bonds, okay? Only single carbon to carbon bonds. Most of saturated fatty acids are solid at room temperature. So, and then most of them are from animal origin. Now, our unsaturated fatty acids have one or more carbon double bonds, okay? So these have double bonds. Most are considered liquid at room temperature, and they have a plant origin. So that's the difference between our saturated fatty acids and our unsaturated fatty acids. So let's take a look at figure 2.13. These are our lipids. So A shows us an example of a saturated, okay? So butter. A saturated fatty acid shows a long carbon chain with a terminal of a COOH group. The other carbon atoms are bonded to the two hydrogen atoms each. Okay, and then let's look at B. That's going to be our olive oil, our unsaturated fatty acid. Okay, so we're going to see some double bonding here. The unsaturated fatty acid shows a long chain, and the other carbon atoms are going to be bonded to two hydrogen atoms each. Okay, remember you have two double bonds that are going to be present within the chain between your adjacent carbon atoms. So that's what makes it different. The difference between our saturated fatty acids and our unsaturated fatty acids. All right, figure 2.14. Excuse me. This structure is going to shows us a structure of a glycerol portion of the left extending from top to bottom, while there are three chains. Okay, so you have your glycerol portion of a lipid, and then you have your three fatty acid chains. Your fatty acid chain. These chains are going to represent our fatty acids. The bottom most chain is going to show a bend. Okay, that bend is because two carbons are double bonded, and they are positioned at the center of the chain. Okay, so that's where you have a bend. You have one double bond here. So as its name suggests, we're going to talk a little bit more about phospholipids. Remember, a phospholipid is a bond between a glycerol component of a lipid and a phosphorus molecule. In fact, our phospholipids are similar in structure to our triglycerides. However, instead of having three fatty acid chains, a phospholipid is generated by a diglyceride. So that's going to be a glycerol with just two fatty acid chains. So our triglyceride has three fatty acid chains and our phospholipid has two. The third binding site on the glycerol is taken up by the phosphate group, which in turn is going to be attracted to a polar head on the region of a molecule. Now, a steroid compound, which we sometimes refer to as a sterol, has its foundations as a set of four hydrocarbon rings bonded to a variety of other atoms and molecules. So although both plants and animals are going to synthesize our sterols, the types that make up the most important contribution to the human structure and function is what we refer to, as we said before, cholesterol. Cholesterol is the building blocks of many hormones within our body. They help with signaling molecules that the body releases and regulates at distant sites. And just like our phospholipids, our cholesterol molecules are going to be found within the cell membrane. Like I stated before, this is where their hydrophobic and hydrophilic regions help to regulate the flow of our substances in and out of the cell. So that is where they are a component or very important in our cell membranes. So part A, look at this triglycerol molecule. You're going to see that it shows a glycerol portion. Okay, this is that glycerol portion attached to three fatty acids, 1, 2, and 3. Now B shows a phospholipid molecule a phospholipid molecule that shows a glycerol portion attached to two fatty acid chains. And though it doesn't have a third fatty acid chain, instead it has a phosphate group, okay, a phosphate group bonded there. And then C is just a schematic representation of a phospholipid molecule that shows your water-soluble head, okay, your hydrophilic head, and your hydrophobic or water-insoluble tail. Okay, this is a phospholipid molecule. And as we go through the chapters, you're going to see us talk about the cell membrane and how these are arranged in order to keep things in or out of the cell. Okay, you have water-loving head with water-fearing or disliking tail. Let's look at the general structure of a steroid molecule now. Now, steroids show a chain of 4 fused rings. The first 3 are a 6-carbon ring, and the last ring has a 5-carbon ring, okay? So you have your 6-carbon rings here, and then your 5-carbon ring here. And then this part B, is going to be the structure of a cholesterol, and it shows us that it has a first ring that is bonded to an OH group, and the first and second rings that are sharing a CH3 group, okay? So this is just showing the different structures of our organic substances. So now we're going to talk about our proteins. A protein is considered an organic molecule. It is composed of amino acids that are linked by peptide bonds. Proteins are also components of many of the body's functional chemicals, including our digestive enzymes in the digestive tract, our antibodies, our neurotransmitters that our neurons are going to use to help communicate with other cells, and the peptide-based hormones that regulate certain body functions, for instance, our growth hormone. So proteins are polymers, and they are made up of nitrogen-containing monomers, which we refer to as amino acids. So our amino acids make up our proteins, okay? Our proteins are formed from amino acids. An amino acid is a molecule that is composed of an amino group and a carboxyl group, together with a variable side chain. Now, there are just 20 different amino acids that contribute to nearly all of the thousands of different proteins that are important to the human structure and the function of it. The unique bond that holds our amino acids together is called a peptide bond. A peptide bond is a covalent bond, so that's that sharing bond. It's a rather strong bond between two amino acids, okay, that is going to form a dehydration synthesis. So a peptide, in fact, is a very short chain of amino acids. Very important that you understand proteins. So let's look at figure 2.17. 2.17, part A is just going to be a general structure of an amino acid. The CH group is bonded to an R group. The portion is common to all of our amino acids, is going to be within the oval. Okay, so within this oval is very common in all of our amino acids. Part B is a type of amino acid that we refer to as cysteine. This is going to be, it has the common group here. And it's going to vary, okay? It's going to vary with this portion, Now, C is another type of amino acid. As you can see, this is very common within the oval, but this group is what makes it different. As you can see here in figure 2.18, you have different amino acids that are going to join together to form peptides, polypeptides, or our proteins. And remember, the bonds between these amino acids are what we refer to as a peptide bond. So highlighted in red, that would be a peptide bond. Alrighty, so a protein shape is essential to its function. Very important that we understand the shapes. A protein shape is determined most fundamentally by the sequence of the amino acids. So how your amino acids are arranged is going to tell us the protein shape. There are 4 levels of protein structure that you should know. You have primary, secondary, tertiary, and quaternary. The primary structure is the sequence of amino acids that is made up of a polypeptide chain, okay? So it is a straight amino acid sequence, okay? A straight line. B Okay, so the second level is going to be our secondary protein structure. The secondary structure, which can take the form of a alpha helix or a beta pleated sheet, those are the two types, is the secondary structure, which can be maintained by hydrogen bonds between our amino acids in different regions of the original polypeptide chain. So you have these hydrogen bonds that are going to help join adjacent and non-adjacent amino acids. The third level is going to be our tertiary structure. This occurs as a result of further folding and bonding of our polypeptide chain. So it's going to be a three-dimensional folded shape, okay? And that's how we determine what the tertiary structure is. Now, quaternary is going to be the fourth level. The quaternary structure occurs as a result of the interaction between two or more tertiary structures, okay? So it's going to be the most complex structure of the four. Now, when protein structures are exposed to extreme heat, bases, or certain other substances, proteins will do what we call denature. Denaturation is going to be a change in the structure of a molecule through physical or chemical means. Denatured proteins lose their functional shape and are no longer able to carry out their jobs. So an everyday example of protein denaturation is going to be curdling of milk. So that's when acidic lemon juice is added to milk, it begins to curdle. It denaturizes the proteins within the milk. In figure 2.19, if you go through them, A, B, C, and D, you can look at the different structures of the levels of your protein structures. A would be your primary structure. B will be the two types of secondary structure. Remember, you have the beta pleated sheets and the alpha helix. So those are going to be the two types of secondary structures that you can have. Tertiary structure, and then your most complex structure is going to be your quaternary structure. All right, so nucleic acids are going to do what we call carrying the genetic code, our DNA, okay? Nucleic acids also aid in protein synthesis, and that is involved in our RNA. Nucleic acids encode amino acid sequences of our proteins. They are very important. A nucleotide is one of the class of organic compounds that are composed of these three subunits. You have a nucleotide consists of a sugar, a pentose sugar, that can either be deoxyribose or ribose. It contains also one or more phosphate group. And then lastly, it contains a nitrogen-containing organic base, okay? And the bases that we know identify are adenine, cytosine, guanine, thymine, and uracil. So nucleotides are formed from our nucleic acids. Okay, everybody got that? Our nucleic acids are formed from our nucleotides. DNA and RNA are made up of nucleotides. Alrighty, so DNA, deoxyribonucleic acid, is a double chain of nucleotides, while our RNA, which we refer to as our ribonucleic acid, is a single chain of nucleotides. So that's how you can tell the difference. Here is what a nucleotide looks like. Remember, the building blocks of all nucleotides are going to contain one or more phosphate groups indicated by a P, a pentose sugar, which is the green S here, and then as well as one nitrogen-containing base, which is the B here. So this is going to show you the structure of RNA versus DNA. The structure of RNA, indicated in part A, shows a chain of nucleotides, each composed of a phosphate group, a sugar, and a base, okay, like adenine, uracil, cytosine, and guanine. Okay, those are going to be the bases that we use for RNA. While DNA, you'll see two chains. Remember, there are going to be a double chain of nucleotides when you're referring to DNA. DNA contains hydrogen bonds that help bring those two chains together. So each DNA strand shows a chain of nucleotides. Each are composed of a phosphate group, a sugar, and a base. And the bases for DNA are our adenine, thymine, cytosine, and guanine. And for DNA, when you are pairing them, adenine is always going to pair with thiamine. So A and T will always bind together, A and T. While C, cytosine and guanine are always going to pair to one another when they are binding. Okay, so that is very important that you understand the base pairs between DNA and RNA and which ones pair with which. They're never going to be a C binding with a T. It's always C and G, A and T. So there are two major types of nucleotides like we discussed. That is DNA and RNA. Deoxyribonucleic acid is DNA and our ribonucleic acid is our RNA. These nucleic acids differ in their type of pentose sugar, okay, which is why their names make them different. Deoxyribonucleic acid is a nucleotide that stores our genetic code, our genetic information. DNA contains the sugar deoxyribose, plus one phosphate group and one nitrogen containing base. Remember, our choices for bases when we are talking about DNA are adenine, that's A, cytosine, that's C, guanine, G, and thiamine, T. The structure of DNA is going to twist slightly, okay? And it's going to form a shape that we know as the double helix. That is the structure of DNA. Now, RNA. Lastly, RNA, ribonucleic acid, is a ribose-containing nucleotide that helps manifest our genetic code as a protein, okay? It aids in protein production. RNA contains a sugar that we refer to as ribose, okay? It contains one phosphate group and one nitrogen base containing base. But the difference is the choices for RNA, when we're referring to our nucleotide bases, for RNA are going to be our A, adenine, C, cytosine, G, guanine, and U, uracil, okay? Uracil is only found in RNA. You will not find it in DNA. While thiamine is only found in DNA. Okay, so make sure you know which bases go to which. This structure of RNA is a single-stranded structure. Single-stranded structure. So you will not see it in the form of a double helix. And that concludes our lecture for Chapter 2. If you have any questions, please feel free to email me. And as a reminder, you can always send your availability to me in order to set up a one-on-one session to discuss any questions you have about this PowerPoint. Thank you.